Today's lesson is going to be about something called Sequential Comparison Index. It is a tool that field biologists use to measure species diversity, basically seeing how diverse a population is, how many different species are available in the environment, and using that as an indicator of ecosystem health. So here we go. So why would we want to measure biodiversity or the diversity of a population? The biggest reason is because the higher the diversity in an ecosystem, the diversity of species that are present in that ecosystem, the healthier that ecosystem is going to be. It's going to be more resilient to disturbances. If there is a disease that wipes through a population, if there's more diversity, it's more likely that something will be more resistant to that disease or disturbance and it wouldn't get harmed as much. So you also want to have, remember, food web interactions, so things that other species can eat, food sources for other species, prey and predator interactions. The more complex those interactions are, the more available food sources that an organism has. So, and basically, whenever an ecosystem is healthier, it can provide better ecosystem services to us. So cleaner air, cleaner water, better recreation, better food production. So healthier ecosystem leads to more diversity leads to a better ecosystem overall. If we've got really healthy soil because of the biodiversity of the microorganisms found in the soil, you're going to be able to grow food more efficiently, things like that. So it does matter the diversity that is in an ecosystem. And so we're going to talk about how we measure that in a stream ecosystem. So how do scientists measure diversity? Well, usually it involves a lot of calculations, very complicated equations, where you look at the relative abundance of each different species in an ecosystem. But that also requires that you can identify every single species in an ecosystem. It takes a lot of expertise, it takes a lot of time, and again, you need to do some math. So in 1968, a non-biologist came up with a way to measure diversity that does not require identification of every single species in the sample. So it saves a lot of time, and it means that someone without extensive training on how to identify species can still get a general idea for the diversity of a population in an ecosystem. So that is the process that we are going to learn about today. It's called Sequential Comparison Index. And it does not require that you are able to identify every species, only that you can tell that one species is different than another. So you just have to be able to tell the differences between species, not exactly what they are. So how do you actually do a Sequential Comparison Index, or SCI? Well, first of all, you have to go out and sample an area. So pretend that we did a dip net and we caught a certain number of organisms, and you wrote down how many of each organism you found. And then you bring it back with you. You have to assign each organism a different color bead. So again, you don't even need to know what these organisms are. You just need to know that they're different from the other organisms in your sample. So let's say that we found 10 of these little swirly guys, and then we found 7 of these little wormy looking guys. And we're going to assign them each a different bead color. So the swirly guys are going to be yellow. The worm looking guys are going to be green beads. Then you've got this circle looking guy, he's going to be red beads, and so on. And you're going to dump them all into a tray, and the tray looks like this. It has horizontal lines drawn across it. So you're going to take the beads that represent your sample and put them in a jar and mix them all up, and then you're going to dump that jar out into a tray that has horizontal lines on it. So that is the first step of the process. You can also use the animals themselves instead of changing them all into beads, but that requires that you take the sample back with you from the stream, and it's better for the health of the stream if you release the organisms that you find after dip netting instead of keeping them, because you don't want to impact the health of the stream by removing the insects that you caught during a dip net. So now, let's say this is our tray. And this is the data that we got after dumping all the beads out, okay? So we're going to compare each animal to the next one in line, moving from left to right on the row. Now, one thing is you want to pretend that each of the lines, that it's not broken up into individual lines, but that the whole thing actually is one big line that keeps going and going and going and going and going and going and going. It just happens to be broken up into these lines for convenience and to save space. So you're going to record how many animals look the same. Again, you don't have to identify it. You don't need to know that the pink beads are dragonflies. You just need to know that the pink beads look different than yellow beads. So here we go. So in the first run, how many beads, how many animals look the same? 2. The next run, we have 3 that look the same as each other. So the next run is 3. You continue this process. There's a run of 1, then a run of 4. Now notice when you get to the end of a line, it's very important that you continue, like going the tray this way, this way, this way, this way. So that's not a run of 1, that's actually a run of 2 because there are 2 animals next to each other if you just made the whole thing into one continuous line. There's the run of 2, then you have a run of 1, and then another run of 1. Okay, so we did 10 runs for this example, but you would continue this process with the whole tray. Just making the run is going to be organisms that look the same as each other. So when we are calculating the SCI, here is our tray of organisms. Here is our data table that we filled out before by looking at how many animals look the same. Now we're going to do our calculation. So the equation for SCI is the total number of runs. So the run number, that's going to be here. The run number over the total number of animals in this case, or if they're being represented by beads, the total number of beads. And you're just going to divide that, and that's it. So it's a very simple equation. So it's the runs over the total animals. That's really important, and I'll tell you about that in a second, the total number of animals that you get. So as you're doing this, you usually do it every 10 animals, so you don't have to do it as many times as I showed here. You only have to do it every 10 animals, but I'm going to do it every 2 runs just to show you how the calculation happens. So in this case, I'm at run 2, and I've had a total of 5 organisms so far. There were 5 beads at the second run. So when I do that division, I get 0.4. Then, couple down, we now are at run 4, and we've had a total of 10 organisms. 2 plus 3 plus 1 plus 4. This is really important that you use the total organisms here. So that bottom number should keep getting bigger as you increase the number of animals that you're looking at. Then in this case, at run 16, I had 13 animals total. At run 8, I had 16 animals total. At run 10, I had 18 animals total. So every time you do the division, you're going to get a decimal that looks like this. Now, a couple things to remember. The SCI is never going to be greater than 1. You're always going to have a number that is less than 1. Never, ever is it going to be greater than 1. So if you get something that's 1, you probably switched, that's greater than 1, you probably switched the denominator and the numerator. Okay, so it's runs, this is the top, this is the numerator, divided by the number of animals total. Okay, so if you get something that is greater than 1, you probably switched those. Okay, so it's runs over organisms. Then to actually figure out what is your SCI, you're going to look at all the numbers you calculated in your table and find the first time that you got 3 numbers that were pretty close to each other. And then, so you get 3, like, oh, they were only a couple decimals apart, 1, 2, 3, and they were all pretty similar. You're going to use the last number that you calculated, and that becomes your SCI. So in this case, the SCI that we calculated would be 0.46 because 0.4, 0.4, and 0.46 are pretty close to each other. It then starts getting further away, 0.5, 0.56. So as soon as you get 3 in a row that are approximately the same, you use the last one, and that is going to be your SCI. The closer number is to 1, the higher diversity. So if you have a 0.83, that's a really high diversity. That means that most of the organisms in the tray were different from each other. If you get a low number for the SCI, that means you had a really low diversity. That means a lot of the organisms were the same as you went through the tray. So the closer number it is to 1, the higher diversity. Now, what you're going to do for today's assignment, you are going to be actually practicing calculating the species comparison index with 3 sets of sample data from 3 imaginary streams that we gave you the data for. So you're going to get a picture that looks like this. And in this case, we used pictures of the actual organisms to make it easier for you and clearer for you. But remember, we'd probably be releasing these organisms into the stream and using beads or something to represent these organisms if we were actually doing it at school. But for now, just look at the pictures. So because of your identification skills, you know that these are scud. But you don't need to know that to be able to do SCI. All you need to be able to tell is that there are 3 of them that look the same. So in this case, you have a run of 3, a run of 1, a run of 1, a run of 1, a run of 1, a run of 1. And remember, it keeps going. There's a run of 2, a run of 1, a run of 1. And look at this. There's a run of 6. Crazy. Okay, so I just did a bunch of the first sample for you in terms of filling out your data. Remember to check the SCI approximately every 10 organisms. If it's 9, if it's 11, that's fine. Just check it a couple times while you're doing it. Check what that decimal would be for the SCI. Remember, you look for 3 places where it was pretty much the same, and you use the final calculation as your answer for your SCI. You're going to record your SCI at the bottom of each page for each of your 3 samples, and then post the work according to your teacher's directions. If you need any help, remember that we are available through office hours for any questions or clarification. Okay, bye.